All right. Good morning. Welcome to the Nature Research Center. So I'm so glad we have a good big crowd. Glad you could join us. So if this is the first you've been watching A Daily Planet this week, you'll hear that this is a very special week. We have a group of teacher scientists who are going to present their research to you today. And this is a project that's called Students Discover. And so you can look it up online, studentsdiscover.org. And it's a large $7 million National Science Foundation grant that we're a part of here at the museum with many partners, Kenan Fellows Institute, Science House, Friday Institute, Grant Pros, a number of agents. But basically what it does is it funds 5 postdoctoral scientists, so early career scientists, who do work in our academic labs here. And these are scientists who are looking for academic positions. They're publishing the results of this work, but they're actually teaming up with middle school teachers. And so each year, each project gets 3 middle school teachers, and these teachers spend 3 weeks down here. Most of them are from, they're from all over the state. They're put up in hotels, and they spend an intensive 3 weeks here co-creating the science with the scientists. They come in as teachers, but they leave as scientists. And they also use those scientific projects to create lesson plans that they're going to use with their students in their middle school classrooms to engage the students in that real hands-on science and to share with them that fun of discovery so that the kids know what it's like to be scientists. And so Monday we heard from Team Soil. Give it up. Where's Team Soil? Woo! All right. Yesterday we heard from Team Ant. Let's hear it. Woo! Tomorrow's going to be the Dino Stars. Woo! But today we have a super popular project called eMammal, and we have our teacher scientists who are going to share with you that project and how they're going to take it into their classroom. So here you go, eMammal.
Thank you, Julie. My name is Nicole Cesari, and I am a 6th grade science teacher here in Raleigh at Ligon Middle School.
I am Lena Deskins, and I have a beef to pick because I'm tired this week of hearing about all these middle school teachers because I am the oddball of the group. I am an elementary school teacher. I teach AIG kindergarten through 5th at Sandy Ridge Elementary in Durham.
And my name is Rachel Palmatier. I teach 8th grade science at Woodlawn Middle School in Alamance County. And we are known as the eMammal team. And for the last 3 weeks, we've been working here at the museum with our own very special scientist, Dr. Stephanie Shuttler. She's right down there in the awesome panda shirt.
So who here has heard of this new movie, The Secret Life of Pets? Came out just Friday, didn't it? So this movie is about these adventures that pets are going on that their owners have no idea. Well, as we started thinking about the eMammal project, I started thinking about how eMammal is all about how we find out about the secret life of mammals.
So one of the first things we have to think about is how we study mammals, or animals in general. How many of you have ever looked at or watched a pet, either yours or somebody else's, before? Raise your hand if you've ever done that. That's probably most of us. That is one way that you can study animals. How many of you have gone out into nature and just observed by listening or looking at the animals out in nature? Also studying animals. How many of you have either read a scientific article or a magazine article or a newspaper article about animals? That is also a way to study animals. For the purposes of the eMammal project, we study animals through pictures taken by camera traps.
So you're probably thinking, okay, well, why cameras? All right, so about 100 years ago, a nature conservationist by the name of George Shiras decided that camera hunting was actually much cooler than gun hunting. So he found out that you could actually study a lot about animals without having to harm them. So what he would do is, if you notice in this awesome picture, his technology is not exactly up to par. Okay, so he had a camera with exploding magnesium powder that was attached to a trap that was then had some bait on the end. So the animal would come by, and then this huge flash would go off, and it would temporarily blind the animals and pretty much terrify them, which gives us an amazing photo like this. As you can see, this really isn't them in their natural habitat. So with eMammal, we want to try to capture them in their natural habitat. So let's fast forward to today and what we are currently using.
So what you see up here and what Rachel is pulling out is called a Reconyx Outdoor Series camera, and these are the camera traps that we use for our research. What's really nice about them, as you can kind of tell, is they're very sturdy so that they can withstand the elements when they're out tied up to a tree somewhere, so not bothered by rain or wind or anything. And they have a heat and motion sensor on them. So the heat and motion sensor is triggered when an animal moves or is in the area, and that trigger sets off the camera, which can then take a burst of 5 sequential photos.
So this process gives us some very, very entertaining photos. We have got to see some really awesome captures this week. And that's all well and good, but this is actual science.
So every time our camera takes a photo, it looks like this. And if you notice in the upper left-hand corner, you'll notice we have 2 red arrows pointing to our date and our time. This is actually really important because as long as we know our coordinates, our GPS coordinates, then this is actual proof that, in fact, this animal was spotted on this date at this time in this area. And scientists use that to help correlate with their data. So, okay, we have these pictures. Wonderful. We have dates and times. Okay, great. Well, how do we actually turn that into science?
So we use a specific software platform, which you can see up here. This is our eMammal software. And once we have collected the camera cards from the camera traps, we put them in the computer, upload all of the pictures. So the pictures themselves are used as data. And as Rachel was explaining earlier, we use the date and the time. Those are very important pieces of data for various research topics that we have. And as the scientist, either in the lab or as a citizen scientist, what you are tasked with doing once you upload those photos is identifying the animals in the picture, which is actually much more difficult than it may seem because sometimes they're camouflaged, sometimes they're moving very quickly so that you can't actually figure out, is that a fox or is that a coyote? And then you just also put in the number of animals that you find in each picture as well.
So when you upload these as a citizen scientist and you're identifying, just like she said, it's not the easiest thing in the world. It turns out that I have a really hard time telling the difference between a red fox and a gray fox. So to ensure that these identifications are stored properly, these next go to an expert review panel. So the expert review takes a look at them, makes sure that the identification done by the citizen science is actually accurate. And then they go to the Smithsonian database where they are stored forever. This gives scientists the opportunity in the future to actually use this data to compare things like global climate change and its effects on mammals.
So one thing that's really, really cool about eMammal is that it's global. If you look at our ginormous world map up here, everywhere you see a red dot are where photos have been uploaded from. And what's cool about that is that different places, that you have a whole bunch of schools, whether it's in Kenya or Mexico or here in North Carolina.
So we have this raw data that is uploaded into the software, but then as you can see here, that data is typically turned into charts or graphs of some sort so that the scientists themselves and specifically student scientists or citizen scientists can see the trends in the data. So we look at things like species richness, which is basically how many different species are there in that particular area, as well as the species diversity and animal activity are just a couple of the things that we look at. So these graphs and charts make it much easier to see those trends and patterns.
As a teacher, it's very important to me that my students' work is actually able to be observed by others. And through this citizen science project, there is some awesome observation going on. So when this is all uploaded, scientists can use this data in their charts and graphs, and then it can actually go into scientific journals. So this here is an example that Dr. Roland Kays, who is the director of the Biodiversity Lab here, he produced this using camera trap data done by students. So your work can end up in a journal. Wow, how cool is that?
So it's very important that schools are participating in this project for a number of reasons. First is that we just need the manpower, essentially, of lots of people collecting data and uploading data and interpreting the data. It's also important because of the research questions that Dr. Stephanie Shuttler has. Specifically, she's looking at the biodiversity of mammals within North Carolina, as well as how animal activity and human activity are intertwined. So we, this year, will be putting camera traps out at Ligon Middle School here in Raleigh, at Sandy Ridge in Durham, and at Woodlawn in Alamance County, which is all the three schools that we teach at.
So in the past, Dr. Stephanie has had camera traps up at various locations, even around the world, and so that is where this storehouse of data is coming from.
So one thing that's been really cool for us as teachers that we've gotten to experience, if you noticed when we had our last slide up, was a little cotton rat in the left-hand corner. Unfortunately, our cameras don't trap anything much bigger than a chipmunk. So we, as teachers, got the opportunity to actually go out to Prairie Ridge at 5 in the morning. Yep, that's before most of you even thought about getting up and having your coffee. Because we had to get out there before these, the sun got out, and we don't want these little guys to get too hot. So we actually got to do some small mammal live trapping out there. And they are doing a capture, recapture research out there. So we would go and we would open up these traps not knowing what type of small mammals would be in there. We did just get cotton rats. And if they weren't already tagged with their ear bling, is what they like to call it, it's just a little number, we actually got to participate and tag those. And it's pretty much just like getting your ear pierced. And then we would release them. And it was just a really, really cool experience that we as the fellows got to experience.
So unless you're like one of Dr. Stephanie's friend and a wonderful Saturday morning is sitting and looking at an Excel spreadsheet, you might not appreciate the joy of data and exploring it and applying it. But that's some of the things that we've been doing this week. And it's been really fun to look at how we're going to take this and use it in our classrooms and get our students excited about it.
As you can probably guess, one of the biggest tasks that we've had over these past couple of weeks has been to actually create lesson plans that incorporate the eMammal data and encourage students and teachers everywhere to participate in the eMammal program. So we have developed several lesson plans involving the data that's already there, but one of my favorite lesson plans as a teacher and as a learner is actually a new experiment that we're kind of kick-starting. It's called the experiment is called a novel object test, which you can see Rachel has our novel object, Fancy the Flamingo. Basically, the purpose of this is you start by just collecting data about what happens regularly with those animals in that environment for about 3 weeks. Then we put the novel object. So we chose this because we figured most animals probably haven't encountered this in their regular everyday life. So we put it out there to see what's going to happen with the animal. And the really cool thing I love about it as a teacher, and I think I would have loved about it as a student, is nobody knows what's going to happen. So as a science teacher, a lot of times I do labs and experiments in my classroom, and I'm like, let's see what's going to happen. It'll be so cool. But in reality, I actually know exactly what's going to happen and how we're going to get there. So as a student, I think it'll be cool to have the experience of your teacher not knowing what's going to happen. As a teacher and a scientist, I'm excited to do something new that I don't know the outcome already. And also cool for Stephanie because she's getting people to help her out with her project. And she also has no idea what's going to happen. So it'll be a really fun experiment. And doing meaningful, real science, I think, will be beneficial for my students as well.
I think one of the things that I want to add before we move on is that I don't know if you guys realize this or not, but scientists, they don't always have the answers, and that's one of the things that we've really enjoyed experiencing here at the museum this 3 weeks.
And finally, we have our lovely pictures of why eMammal is so awesome, and that's because you can participate. You can become the scientist.
So if you're a student out there who's thinking, this sounds totally awesome, I want you to go to a teacher and I want you to say, citizen science, students discover, eMammal, let's do it this year. If you're someone who wants to volunteer to put a camera trap out in your yard, I want you to contact Dr. Stephanie Shetler here at the museum and set up a camera trap so that you can find out the secret life of your backyard or schoolyard. So do we have any questions?
And I just want to say, this is a lot fuzzier and cuter than the little ants. I'm just saying. But we love ants.
Thanks, sir. Yay. Do we have any questions for our team?
What has been the coolest thing you guys have seen on the camera trap so far?
I would have to say that we did some uploading from here locally and seeing that there is actually like coyotes and deer and squirrels and all that you'd expect to see, but the coyote kind of surprised us right here in Cary. And then looking at some of the South American, we saw some awesome ocelot and anteaters.
I actually have two favorite. One of them was I got some really cool like puma shots, and he was right in the camera, and he just the expression on his face was really cool. But then with our Cary photos, we got to see a fawn through this set of photos, because every time we take photos, it takes 5. We actually got to see a fawn nursing with its mom and then playing around the pink flamingo, which was kind of funny. He just kind of hopped around it.
Kind of piggybacking off of what Rachel was saying, I liked seeing when you could tell that it was that animal's home or its habitat. So like with the deer and its fawn, you could tell that that was their living space, which was really cool to see like their more regular interactions. And I also had several pictures from other countries that the camouflage with animals was insane. I had one picture that there was a monkey, and it took me so long. I was like, I know there must be something here because it triggered the camera. And so playing almost like a Where's Waldo kind of thing, trying to find the animal was really cool. And then once I found it, it was just its tail that had triggered the camera. And so I had to spot just its tiny little tail in amongst all the leaves.
So early in the presentation, you guys mentioned the scientist who had the big exploding flash. Do the camera traps that you used, is there a visible flash with those?
They do not have a visible flash.
So how do they do the night photography?
Using infrared technology, yeah.
So what kind of concrete data are you guys kind of gathering? Is it mostly like migration patterns and like sort of habitation patterns, or is it like behavior, or like how is it kind of quantified, I guess?
So the specific data that we're collecting from the photos is the photo and what animal is in it specifically and the count of the photos. And then as you saw in one of the pictures, the date and time. From that is how you have to analyze the data to see those other things, such as the animal activity or migration patterns or whatever. That's not concrete data that we're directly extracting from the pictures themselves.
And what's cool though is on our actual eMammal website is they've already processed through a lot of the data that shows species richness, species diversity, those different things. They're actually starting to be able to track different patterns of where, like for example, coyotes are really big. They're trying to figure out, we learned today that coyotes have actually only been in North Carolina for 30 years. So they're curious as to where they're located and why they're located there. Other stuff?
So essentially what happens are the photos turn into numbers, and that roughly correlates to abundance. So you can look at North or Raleigh area, and you can see I have more detections of coyotes at this site and less at this site. So we think there's a higher population at this site. So it's numbers and looking at the distribution over the entire area. And then they mentioned activity patterns, so you can group the counts by time and see if an animal is nocturnal or not. And what we eventually do is relate it to human patterns or other things that we think might be driving the differences. So the environmental factors like habitat or human development might be changing the activity patterns in animals or causing them to live in different places. But we also do use behavioral data too. So another thing that I worked on with deer, actually, perfect timing, is we had a study across the eastern United States in different parks where they were hunted, and there's hiking in all of the parks, and we looked to see if deer change their behavior in relation to hunting and hiking. So if a deer is really scared, it's probably going to be scanning the environment looking for predators a lot. But if it's not scared, then it's going to spend its time eating because it's better for them to get fat and they can have more babies. So I actually counted the head position, myself and interns, we counted the head position of thousands of deer in these photos, and then we related it to coyotes and human hunting and human hiking. And it turns out that deer really are not that scared of coyotes and both human hunters. So with the coyotes, we think that they're not really hunting adults and only taking fawns. And with the hunters, we're not sure exactly what's going on. Maybe they leave the area or become more nocturnal when hunting's not active. So all different types of stuff that we can do with the data.
I have a question. Just because you're taking, the cameras take images in 5 photo bursts, when in this kind of situation, is it just the continued movement that continues to trigger 5-second bursts or 5 image bursts?
Yes. So some of these are pictures turned into movies, so it looks like a movie. This is actually a video, though. So you can set the camera trap to a video recording. I think that one's pictures. So you can set the camera trap to a video recording and actually get live video feed for the animals.
This sounds so exciting, and I can imagine it would be really fun and kind of different for kids in classrooms to do. I know one of the things that's a big issue for teachers is the standards that you have to meet and requirements about what students need to learn in each grade at each grade level. Can you talk a little bit about how, in your view, participating in a project like this might help kids gain some of the skills that they're required by the state to know?
Absolutely. One of the lesson plans that we created, in my teaching of AIG students, I teach literacy and math. So I'm really trying to take the science and cram it right in there to those areas. So we looked at biomes and related what we have as data to the different biomes. So the students will not only read and learn about the biomes, but they'll also use the species diversity maps and things like that to help them relate what they learn about biomes to the eMammal data as well.
Well, I mean, that's one of our things that we have to do when developing the lesson plans is we specifically hit state standards, but also the new generation of or next generation, of scientific standards. What's been really cool though, and what's kind of different about our particular group is because we have Lena, we're not just hitting your middle school levels, we're hitting some elementary too, which is the first time, at least we've seen in terms of people creating lesson plans for a younger age group. So what's interesting is we have one lesson, but then we have different forms or different things like that depending on the class. So we have another one with the flamingo where it's for 7th, 8th, and 4th grade. We're hitting all of those standards.
So kind of like Rachel was saying, we have created 3 different lesson plans, and all of those are standards-based. And what we've tried to do, because Rachel is 8th grade, Lena teaches elementary, and I'm 6th grade, is at the very least we've tried to make sure that each of those lesson plans hit standards for each of those 3 grade levels. So we've been working on incorporating some differentiation, even if it's with the questioning or what have you within the lesson, that kind of can gear it towards different grade levels and different standards within those grade levels as well.
Any other questions?
So what do you think the impact would be for the students to see what wildlife lives on their campus or around their home? What's that mean to them?
For my students, I think it'll be incredibly fascinating because I have a large neighborhood population, so my school is right within the neighborhood that a lot of my students live in, and I don't think they realize that there are animals, especially because I'm in a little bit more of an urban setting because I'm just right downtown. So I think it'll be really cool for them to see that, and I'm also looking forward to getting the opportunity to actually take them out into nature because a lot of them are used to like more of the concrete jungle than the actual like woods and the forests and stuff. So I'll be interested, if nothing else, just to get them outside and interacting with nature, which I don't think is something they're accustomed to.
In that same regard, it really had us looking at our school grounds and thinking about great placement for this and great places to get the data that we're hoping for. And so it's nice to get the students thinking about that with us.
I think it's going to make them really excited, even if they see a deer, because if you could have seen the three of us looking at the pictures from Carrie, and Stephanie was sitting there looking at us, and we're like, it's so cute. Like, she just started laughing because she's like, oh, I see these all the time. But we're like, I know, but it's a baby deer, you know? So I really think that the kids are going to be surprised to see those things, but also to know that they are in their school. You know, this is where you play baseball at, or this is where you walk with your parents, you know, but it's got a whole nother secret life that you don't know about. And I was actually fortunate enough that Stephanie had put up some cameras at the end of this school year, and my students that are now going to be ninth graders, some of them have actually emailed me like, hey, Miss P, what'd you get on the camera? Did you check the camera? And I'm like, not yet, guys. We still got to wait. So even kids that aren't going to be participating this year in my class were excited because they saw it get set up last year.
Some of the things I've heard from teachers at other schools are that a few things is that it reaches students who traditionally don't do well or are typically not interested maybe in other classroom activities. Some of the more middle of the road students really took interest and got really excited about the camera traps. Even if the students didn't get anything exciting, like even domestic cats, what they noticed is that the animals acted differently from one another. They noticed some different things that the domestic cats were doing that the wild animals weren't doing. And they also realized that animals are not just robots, that they're going through their environment and they're making decisions and not just randomly going places. And then finally, I can't emphasize Nicole's point enough of just going outside. So we have kids running camera traps all over the world. And some of these kids are in really rural India where it seems like outside is like really accessible to them. And even the teachers there were like, the kids got so excited to just go outside and set up a camera trap. So just that simple act, which I didn't realize was such a big deal, was just really exciting and interesting to them.
Any other questions? Awesome job, you guys. Thank you so much. Let's applaud again for eMammal. Thank you.